So welcome everybody to today's edition of our webinar on emerging topics in biomolecular magnetic resonance. Both speakers agreed to recording today, so we are currently recording and we will upload the recordings to the ISMRBS YouTube channel. Please don't forget, if you have any questions either during the talk or at the end, please use the Q&A box. Today we have two fantastic presentations. The first one will be by Roberto Salinas from University of Sao Paulo, and the second one from Anthony Bishop, and now at Texas AM, if I understand this correctly. With that, I hand over to Rob Kaptein, who agreed to introduce Roberto.
Okay, it's a great pleasure to introduce this first speaker today, Roberto Salinas. Welcome, Roberto. We know each other already for more than 20 years since you started your PhD research in Utrecht with Rob Boelens and myself on lacquer pressure DNA interactions. What else? You know? So it's good to see you again. Roberto did his bachelor and master studies at the University of Sao Paulo in Brazil. He then got a scholarship to come to Utrecht for his PhD, which he finished in 2005. Afterwards, he went back to Sao Paulo, where he is now professor of biophysics, I believe. His research is on structural biology of systems of medical interest, mostly using NMR spectroscopy. I keep it short. The title of his talk is The Dynamics of Calcium Binding Domains of the Sodium-Calcium Exchanger Investigated by Solution NMR Spectroscopy. Sorry for the slight modification. But, Roberto, the floor is yours.
Okay. Thanks, Rob, for this introduction. It's nice to see you again. I'm going to share my screen. Let's see. Okay. Let's see. Can I? So can you see my screen?
Yes.
Right. So thanks for this invitation and for the introduction. I'm going to speak about the work we have been doing in the past few years in Sao Paulo. It's basically something that I started when I did a postdoctorate with Rafael Bruchweiler at Florida State University, and then back in Sao Paulo, I continued to work on this system. And it's basically on the sodium-calcium exchanger, and with a focus on the calcium binding domains. So cells, just as an introduction, cells have different mechanisms to control the intracellular calcium concentration. So there is the ATPases, which are membrane proteins that basically hydrolyze ATP to export calcium against the gradient, so to remove calcium from the cell. There are calcium channels, and there are also exchangers. The sodium-calcium exchanger particularly is a membrane protein that uses the sodium electrochemical gradient to pump calcium out of the cell in its major transport mode. So the exchanger, as you see, it's going to exchange basically 3 sodium ions in exchange for 1 calcium. It is the major protein that is involved in the maintenance of intracellular calcium homeostasis, particularly in excitable cells. It's involved in cardiac diseases, but also in neurodegenerative diseases, and also in cancer. As you see, the exchanger has a topology. It's a very large membrane protein with 10 transmembrane helices that basically contains the sites to bind sodium and calcium across the bilayer. I hope you see my mouse here. So here's this crystal structure. There are a couple of crystal structures of the prokaryotic exchanger. So the eukaryotic ones have never been crystallized. And in the prokaryotic exchanger, basically you see the 10 helices bundle. Here with sodium, you see the sodium is bound, and here with calcium. And it basically transports in a kind of constitutive way. Usually these sort of exchangers, they cycle between conformations that expose the binding sites to the outside of the cell, and then to inside of the cell, and then they're going to bind calcium and then change conformation, then exchange the substrate and exchange the conformation again, and then they cycle through these outward facing and inward facing conformations as they bind the substrates. Now, but the interesting thing is that the eukaryotic ones, they have a very large intracellular loop that you don't see in the prokaryotic ones. And that means that they are regulated. So they're not working all the time. And basically what you see is shown in this scheme of a patch clamp experiment. So if there is sodium and there is intracellular calcium, then the current through the exchanger peaks, and then it goes down slowly through a steady state current. Now, usually there are the mammalian exchangers that have two calcium binding domains, which are called CBD1 and CBD2, which are located in this loop. So they are intracellular domains, and they're going to bind the regulatory calcium. So somehow calcium binds to CBD1 and CBD2 and activates the exchanger. So I have been studying the Drosophila exchanger, which works in the opposite way. So it binds, CBD1 binds calcium, and the exchanger is inhibited, and you see it as a decrease of the peak current and also decrease of the steady state current. So one of the things we are interested in is basically understanding how that happens. So how the calcium binds far away from the transport sites in the membrane is able to regulate the exchanger activity. Now, the calcium binding domains, they are basically two, they have immunoglobulin folds, so they are basically a beta sandwich, and calcium binds on the loops on the top of the beta sandwich. So this is the crystal structure of CBD1 that binds 4 calcium ions, and this is the crystal structure of CBD2 that apparently doesn't bind calcium at physiological concentrations in the case of the Drosophila exchanger. Furthermore, there are different isoforms due to splicing regulation, and the result is that the regulatory properties of the exchangers are a little bit different. And these isoforms, in the case of the Drosophila exchanger, basically they are located in the CBD2 domain, where this alpha helix in this loop that's called FG loop, the region in red, it has a little bit different amino acid sequence. So just 5 amino acids change, and then the 1.2 isoform is less sensitive to the calcium regulation than the 1.1 isoform. That's the consequence. Now we have been working with the 2 domains separating solution. They are relatively small for NMR, so we could assign, and this is what you see is the CBD2. So we measured heteronuclear NOEs and also R1 and R2 for N15, and we could calculate in other parameters. And basically what you see is that the loops at one end of the domain, they are much more mobile than the beta sandwich core. And this is, so this is for the isoform 1.1. So here are order parameters, amide order parameters mapped on the structure, also for the 1.2 isoform. And what you see in gray is basically regions that have not been assigned, either because the peaks are missing or because correlations are missing. But this pattern is typical for these kind of domains, and also same thing was seen in the mammalian exchanger for the CBD1 and CBD2 of the mammalian exchangers. Now, if you titrate calcium and record the HSQCs, you see that peaks move in the case of CBD2 in fast exchange. That means that calcium is binding here on the opposite location of the FG loop. And this is where you would expect it to bind the calcium binding site. However, the calcium binding affinity is very, very low. It's in the millimolar range, which means that it's not physiological. Compared to the mammalian exchanger, the CBD2 domain in the mammalian exchanger is called NCX. It binds calcium much more tightly. And the reason for the low affinity of the Drosophila domain, we think it's because this glutamic acid 551 was changed for a lysine. And also this glutamic acid was changed for a serine, and as a result, the affinity decreases significantly. So the binding of calcium to CBD2 is not physiologically relevant in the case of the Drosophila exchange, which is also called CalX. Now, we wanted to understand what was the orientation of this loop. It's called FG loop, and in solution. And for that, we basically measured N15 amide RDCs, reservoir pore couplings, and this is a plot of calculated versus experimental RDCs. And you see this is a relatively high Q factor, but these are for all amides. If we basically use only the ones which are located in secondary structure, then the Q factor decreases. And we calculate the alignment tensor only on the beta sandwich and only on the FG loop. And we observed similar overall alignments, which is the AZZ, and which are a little bit smaller for the FG loop. It seems that there is some mobility, but not much. This is the correlation coefficient between the two alignment tensors, which suggests that they're relatively collinear. And we also did calculate the molecular dynamics trajectory for 1 microsecond to understand better which kind of movements these helices do. And basically what you see, helices H1 moves up and down, and this is a distribution of the angle, the average angle between the helix axis and the beta sandwich. While the helix H2, it's in the back, I'm not showing, it fluctuates much less than helix H1. Now, what about the other domain? CBD1 domain is the one that binds calcium more tightly. And it's different because it's we couldn't really assign all the residues which are located in this gray region in the absence of calcium in the aqueous state. So it seems that there is some sort of dynamics in the aqueous state, which is in the intermediate time scale, and lines get too broad. But then when you add calcium, then peaks starts to show up. And basically this is a superposition of the spectrum in the aqueous state in blue and in the calcium bound state in red. And you can see that residues, peaks due to residues which are located in the calcium binding region, they basically show up in the calcium bound state. And you don't see intermediates. So we see signals for the aqueous state and for the calcium bound state with all 4 calcium ions bound. So this suggests that the binding is cooperative. Therefore, we plot the intensity of the peaks that are showing up as a function of the calcium concentration, and then you see titration curves. So in black, these are the experimental titration curves, as you see. And sometimes these titration curves are hyperbolic, sometimes they're more sigmoidal. And we could basically explain this assuming or calculating a term due to the conformation, to the chemical exchange contributions to the R2 relaxation. Basically, if you calculate and you use this in the fitting, you can basically explain this sigmoidal profile. And these are fittings to the Hill equation. So we could basically for several residues, and then we have a distribution of the Hill coefficient, which is 2.9, which is indicating that the bind is cooperative, and then the average the association constant is of the order of 4 micromole. Now, so this is a plot of the correction term that we proposed on the intensity, and that's how it affects the titration curve. So the exchange contributions basically to the R2 relaxation rate are maximum at the middle of the titration, and then they cool down, and then this would cause this distortion in the titration curves. And this factor is going to depend on the difference in chemical shift between the free and bound states, and also on the off rates. So it depends on the combination of this delta omega and on off rates, you are going to get more or less distortions. And in fact, the resins which are close to the calcium binding sites that experience more dynamics in the April state, they showed more distortions in these binding curves. Now, CBD1 and CBD2, they're actually covalent connected, forming a two-domain calcium sensor. That's what you see here. This is a crystal structure. And this two-domain calcium sensor is called CBD1, and it's really the calcium sensor of the Drosophila exchanger. And then what we wanted to know is okay, when CBD1 is connected to CBD2, how is the dynamics between these two domains? We can basically assign the CBD1-2 resonances by comparing with the N15 HSQC spectra of the CBD1 and CBD2, which were already assigned. So most of the assignments were obtained basically by transferring the resonances from one construct to the other. We measured 15 relaxation rates. For now this is for the CBD1 two-domain construct in the April state and in the calcium-bound state, and basically you see a large increase in the R2 over R1 ratios, which is consistent with a molecule that's becoming larger. Basically that's what you see. So you see an increase in R2 with the decrease in R1. So this is consistent with an increase in molecular weight, and that cannot be explained by aggregation. So we checked this. So it's basically the molecule is becoming bulkier. We also calculated the ensemble average orientation between the two domains using RDC. So basically measuring RDCs on CBD1-2, fitting on one domain to calculate the alignment tensor, fitting on the other domain to calculate the alignment tensor, and then orienting one domain with respect to the other. This for the calcium-bound state and for the April state. And what we found, basically was in the calcium-bound state, we found basically the same orientation that we see in the crystallographic structure. But then in the April state, we see, we obtained a solution that is not realistic. And the problem here is, of course, is that in the April state, the two domains are flexible with respect to each other. And then this approach doesn't yield a realistic solution. We more recently, then also recorded the SACS measurements on CBD1-2 in the April and in the calcium-bound state. And if you look to the overall, here is the pair distribution function. And it's basically in the April state, it's more consistent with flexibility between the two domains and rigidity in the calcium-bound state. And also here is the scattering profile. We can basically reproduce the scattering profile using the crystal structure in the calcium-bound state, but not in the April state. So this indicates that there is indeed conformation heterogeneity in the April state, while the crystal structure represents well the calcium-bound state of CBD1-2. So the situation we have then is basically CBD1-2 is the two domains are relatively flexible with each other in the April state, but then in the calcium-bound state, calcium-bind stabilizes a rigid orientation. Now we also had done MD simulations of CBD1-2, and if we basically do MD simulations, and we also use the RDCs and calculates a Q factor distributions for all snapshots on MD simulations. We basically see in the April state the structure. This is an R factor distribution. So basically for the April state, the crystal structure does not fit in the RDCs, which is what we expected, and then there is no improvement during the simulations, except for those conformers which are basically on this region of the distribution. And if we look to them, they are a little bit more closed than the crystal structure. So it seems that in the aqueous state, the ensemble is dominated, by more closed conformations. In the calcium, this is the distribution, the R factor distribution for the calcium bound state. You see an improvement for the solutions, for the snapshots in this region of the distribution. And so here I'm showing the one which is indicated by a star here. Basically what you see here, it's an improvement of the overall geometry of the NHs. Now you can then basically use the ensemble optimization method from Paul Bernadotte and Sachs intensities to basically refine this. We chose basically sub-ensemble as input for the EOM to refine and calculate a small ensemble that would be consistent with the Sachs data. And what you see in the crystal, in the case of the calcium bound state, your ensemble is basically well represented by the crystal structure as expected. But in the aqueous state, we basically see that there is it basically selects closed conformations and also open conformations. And then we interpret this as basically an indication that what we have in the aqueous state is that CBD12 samples extended conformation, more closed conformations. It probably spends more time in the closed conformation, the calcium binding, and stabilizes the extended conformation. Now that brings us to a model for how calcium binding to CBD12 is going to inactivate the exchanger. Basically, by stabilizing an extended conformation, it causes a tension that should affect the transmembrane helices, and thereby that should be the way how it triggers calcium inhibition of the exchange. Now, the problem of the splicing then. The Drosophila has two isoforms, 1.1 and 1.2, that differ in 5 residues in this region, which far from the calcium binding sites. The CBD12 constructs of the two isoforms that basically bind calcium with the same affinity. So that doesn't really explain, that doesn't help us to understand how these two isoforms might have different calcium regulation phenotypes, because the 1.1 is much more sensitive to calcium, and the 1.2, which is shown in red, is less sensitive to calcium. Now, when you run MD simulation, these are 1 microsecond MD simulations of CBD12 constructs of the two isoforms. What we see is that because of these amino acid substitutions in these helices, the contacts at the interface are a little bit different. For example, arginine 584, which forms a salt bridge with the aspartate 552, this interaction is typical of the calcium-bound state, and you see it most of the time for the 1.2 isoform, even in the absence of calcium, while you don't see it for the 1.1 isoform. We really never could do NMR on this CBD12 1.2 construct because the lines are relatively broad and it tends to aggregate in the NMR tube, so it's a difficult sample. So what we did was to measure Sachs. And now in contrast with the 1.1 isoform, here the crystallographic structure represents very well the scattering intensity of the aqueous state, of the unbound state. And also the pair distribution functions shows evidence that the two domains are relatively more reached with respect to each other in the 1.2 isoform than in the 1.1 isoform. So overall, what we learned so far is that either calcium binding or substitutions that affected the interdomain contacts at the interface, they're both able to adjust the conformation energy landscape of CBD12, either stabilizing more compact conformations in the 1.1 isoform, even though it still fluctuates between open and closed, or stabilize the more extended conformations in the case of the 1.2 isoform, and this has implications for the mechanism of calcium regulation of this exchanger. Now, with that, I'm closing. And I'd like to change to acknowledge people who have done this work. So this was in collaboration with Rafael Buschweiler. And most of the working done with the CBD1 exchanger was part of the PhD thesis of Laera Bicu, which is now in Switzerland. All the SACS data and SACS data analysis has been done by Maximilian, which was a PhD student in our group in collaboration with Cristiano, which is who is the SACS specialist, and she's now as a postdoctor at the NIH. And all the data done with the 1.2 isoform was done by Mario Andres Rodrigues Pineda, who was my PhD student, and it's still is a postdoctor in Brazil at the moment. And thank you very much for your attention.
We don't hear you. You're muted.
Okay, that should be better. Yeah. Okay. Okay. Let me start the question and answer session. I have a question about the allostery and the mechanism of that. If I understand you correctly, Roberto, you said that the calcium and sodium ions are involved as allosteric effectors, but at the same time, of course, they're substrate of the whole process. Huh? So usually these roles are separate. Right. So this is a bit difficult to understand. Can you explain how that came about?
Yeah, well, I think it's easier to understand the calcium regulation because, for example, with the mammalian exchanger, it's not transporting all the time. And then when intracellular calcium concentration increases, it binds to CBD1 and then activates the exchanger, and then transport starts. So that's the situation for the mammalian exchanger. And basically that means that the exchanger is going to work only after a certain intracellular calcium concentration. This is important for excitable cells because it's basically an important player only when the intracellular concentration goes up, because these cells they have spikes on the intracellular calcium concentration. So at lower concentrations, it doesn't work. Then when the concentrations become very high, then it needs to act basically to remove the cell, to remove calcium. Now in the Drosophila is the other way around because it forces the cells to increase the intracellular calcium concentration. And this, it seems, that has some importance for the in photoreceptor cells. So basically in the vision system. And now the sodium intracellular, the sodium allosteric regulation, it's a bit more difficult to understand, but it seems it has to do with the decay to a steady state level of the ion exchange current. So the currents go up, peaks, and then it goes down to a steady state level. And it has to do with this decrease. But the mechanism, it's unclear. For sodium, it's totally unclear.
Okay, well, it's a very interesting problem you're working on. Let me see if there's one other question by Philip Neudecker. And he asked the question, I'm sorry I missed this. What is the definition of ETA?
ETA?
Yeah, well, that's what he...
ETA, I think it was in your parameter ETA.
Sorry, my parameter ETA is actually...
That's the parameter, sorry.
Sorry, I didn't explain this. I should have explained that. So my parameter ETA is basically the ratio between the peak intensity at any point in the titration and in the plateau. So basically, to analyze the calcium binding curves, basically plot the peak intensity as a function of calcium. And that is actually a ratio between the peak intensity and the plateau. And that's the ETA. Okay, and that depends on the population of the bound state as well, but it's also affected by the relaxation rate. So the peak investors are also dependent on the relaxation rates. And this depends on the intrinsic R2 rates, but also on the exchange rates between free and bound. And that's the problem there.
Okay, is there any question from the panel?
Yes, I would like to ask one. So, Roberto, in your cartoon, there was also these regions which looked like disordered, and where you added pluses and minuses close to the transmembrane part. I mean, what is known about the contribution of these regions to the regulation of the channel?
Yeah, so these regions are important because there is one that I didn't mention, which is positively charged. And if you design a peptide corresponding to the same frequency, it inhibits the exchange. So it believes that it's generally believed that this region binds somewhere, not in CBD1 or CBD2, but in some other place. And that is part of the mechanism of regulation. But these are very difficult regions to study because it's very close to the membrane. All secondary structure predictions point that there is a lot of disorder, so intrinsic disorder propensity. For which value he has expressed some of these regions for the mammalian exchange. It seems that they are disordered indeed. I think until we don't have a full structure of the complete protein, it's going to be difficult to understand what they do actually.
Thank you.
Any others from the panel?
Roberto, when you showed the MD simulations for the helix and the FG loop, the distribution looked kind of two-state-like.
Right.
And if you look at the structures, is it obvious why it kind of latches into two states rather than just being a broad distribution of that loop swinging back and forth? Any hints as to what the molecular interactions are that cause it to be two-state-like?
Right. No, I didn't look to the to possible interactions there that could stabilize these two states, but that's an interesting observation. But I didn't look to these, and it's curious because the other helix, it basically doesn't move so much.
Right.
It's just the first one.
Or you might have expected your first one just to be a very broad distribution.
Right. Right.
But it can't occupy very much the intermediate angle, right? Something's happening.
Yes. Yes, that's an interesting point. I didn't look at that, but it's a good point.
Okay. Any other from the panel? I don't see any other questions from the audience. So if there's no question from the panel, then we can close this session. Roberto, I enjoyed your lecture. I think it's very complicated, but very interesting research. Thank you very much. And it was nice seeing you. And I hope to see you again before not too long in real life. Yeah, thank you.
Thank you.
Okay, yeah.
Ed, would you jump in?
Thanks for the warning about the muting. It's a sad moment. It came to me very much as a surprise that we lost a dear friend of the NMR community. Ray Freeman passed away yesterday. And I was asked just less than a half hour ago by Art to say a few words in memory of Ray, who really is one of the original leaders of modern NMR. So my words are rather unprepared and perhaps rather rambling, but I realize that many of the younger ones in the audience today may not even know who Ray Freeman is, just like you may not know the name of Abraham and some of the others originators of NMR spectroscopy. Ray Freeman was really probably the first to bring modern two-dimensional NMR into the chemistry community. The hardcore foundation, the mathematical foundation, really had been developed by Richard Ernst, but they did this sort of on parallel avenues. And Ray was really the one that translated it into bringing NMR spectroscopy into the normal NMR lab, because his mission really was, and his motto that he taught us as students. I was a graduate student with Ray Freeman back from 1979 to 81. It was that if you can't explain it to a chemist, it's not worth doing. And he really always pushed us on explaining and conveying it in words that the community would understand and appreciate. It really was an incredible experience to be in his group. I had the great fortune of being there at the same time as many of the other current leaders that you guys may consider to be old people, but we consider ourselves young. There was Malcolm Levitt was in the office with me. Jeffrey Bodenhausen had just left the lab when I joined. Gareth Morris, now professor at Manchester, as a leader in small molecule NMR. James Keeler, who many of you may recognize at the University of Cambridge. He became a leader in chemical applications of magnetic resonance. My dear friend Tom Frankiel is still at the MRC, running the labs there. AJ Shaka is a big name in chemistry NMR. Eric Skupse followed after we had left, and many other names. I won't bother you with it, but what I wanted to sort of like exemplify here is that Ray really was the coach, the trainer, the mentor for many of the people that you now recognize as being, perhaps you might think, old folks in NMR. He was a colleague at the same time as Rob Kaptein, and they were dear friends. Both of them were at my PhD defense. So, I mean, it's, for me, losing Ray Freeman is like almost like losing the history of NMR. On the other hand, Ray taught us so many things, and I share so many happy moments from the days that I had the fortune to work with him, that he will be with me forever. I mean, he really was an inspiration to make NMR interesting, to make it fun, and to sort of enjoy science. Those of you who were at the ENCs back in the late 70s and 80s up till the early 1990s, remember that he was always a plenary speaker there together with Richard Ernst. And Ray actually was known almost as much for his science as for his funny cartoon slides that he was able to sneak in there. Some of them wouldn't pass the ethics police anymore these days. The rules were a little bit more liberal back in the 1980s and 70s, but he really had an incredible sense of humor, and he was able to convey that sort of like fun in science to the younger community. And so we'll all miss him, but we will have him in our memories for as long as we live. Those who had the pleasure to know him, and those who didn't know him, you know him indirectly, because many of those who you're meeting today were trained and coached by Ray. So at this point, like I said, I hadn't prepared an eloquent speech for this, but those are my first rambling thoughts on his passing. Thanks very much.
Thank you, Ed, for the words. We have now a second presentation, which is given by Anthony Bishop from Josh Vaughn's group. So Anthony obtained his bachelor from the Rensselaer Polytechnic Institute in bioinformatics and molecular biology, and then actually stayed at the same institute to obtain his PhD in biochemistry and biophysics, I think under the supervision of George Makatadze. I hope I pronounced this correctly. And then he moved on for a postdoctoral stay with Josh Vaughn, where he's working actually on a number of projects, all the way from fragment-based drug discovery using reverse micelle strategies developed in Georges Fran's lab, to characterization of allosteric effects of enzyme ligand binding, and simulation of methyl order parameters for protein ligand interactions. But then there's another fourth project, I guess, which he will tell us today about, which is about developing for NMR. Anthony?
Thank you so much for that wonderful introduction. Let me just share my screen and we'll get underway here. Okay, once again, my name's Anthony. I'm a postdoc in Josh Vaughn's lab, and I'm very excited to tell you about this new automated assignment algorithm for protein backbones that I've devised. So as we all know, the general triple resonance assignment strategy involves various triple resonance experiments that correlate carbon atoms to the amide group. And because each amide group it corresponds to a single amino acid, and all amino acids have an amide group with the exception of proline, we can run all of these experiments and generate what are often termed spin systems, which is the total set of carbon resonances and their attached amide groups. So we get a single spin system associated with each associated with each residue within the protein. And because there's overlap in the resonances, we can then use this overlap in order to link these resonances together and form what are called connectivities. And then these chains will expand, and then they can be assigned to the protein structure or the protein sequence. And while this strategy has been quite successful for smaller proteins, there really hasn't been much progress in the assignment of particularly large proteins. So shown here is the distribution of assigned proteins in the BMRB as of this past October. And you can see that MBP, which was assigned a couple decades ago, there are almost no proteins much bigger than that have been assigned. In fact, fewer than 60 proteins larger than MBP have been assigned. And the reasons why this aren't unknown. There's severely attenuated signals associated with larger proteins due to rapid T2 relaxation, in addition to other unavoidable sample conditions that might be necessary. Not to mention just the increased level of spectral degeneracy that leads to the ambiguity in making those hard connections. So I wanted to sort of tackle this problem because I was given the task of assigning a relatively large protein, and I was running into problems doing it manually. So I went to look at some automated assignment algorithms, and what I noticed is that most automated assignment algorithms that already exist only use BMRB-derived amino acid resonance distributions to try to map the spin system chains to the sequence. And a number of algorithms improve upon this, though, by using actual predicted chemical shift data from a 3D structure, which sort of lessens the ambiguity of that mapping process. But when I was looking at it, I thought that we were overlooking as a field the ability to do something, take that a bit of a step further, and really using predicted and observed chemical shifts in a Bayesian fashion to map the spin systems to the sequence positions. So to this end, I developed an algorithm termed Bellagio, which automates this process of mapping spin systems to residue positions. And it does this via simulated annealing, optimizing a Bayesian pseudo-energy function. And in case if you're curious where the name comes from, it's a portmanteau of Bayes and Bellagio, which is a famous casino in Las Vegas. So simulated annealing is a cross-disciplinary method of optimizing a function to find usually a global minimum in that function's value. And the reason why we need some sort of heuristical algorithm to optimize this problem is that if you were to examine every possible solution, even for a 100 residue protein, there would be roughly 100 factorial different possible solutions. So simulated annealing is a way of really cutting to the solution without having to visit every possible solution along the way. I won't go into it a bit, in addition, simulated annealing is not new to assignment algorithms. It's actually been used before in at least one algorithm that I know of called Monty, which was developed by the Rule Lab about 20 years ago. So our method improves upon this, one, in terms of the energy function, but also in terms of the fact that we use an on-the-fly annealing schedule, which has minimal user intervention. I won't go into too much detail about that, but you can ask me about it later. So just as a quick overview of how simulated annealing works, if you were to plot all of the possible solutions and the associated calculated energy, which is determined by your energy function, your goal is to find the lowest energy state, and you devise your energy function in a way such that the lowest energy state is the solution that you're looking for. You first begin by picking a random solution, and then you make small changes to that solution and calculate the change in energy associated with that change to your solution. You then apply the metropolis criterion, and which is a function of the minimum of 1 and e to the negative delta e over t. And you only accept it with the probability governed by this calculated value. So as a result, if you decrease in energy, this equation states that you always accept the swap. But if you increase in energy, the probability that you accept the swap scales with the energy increase. So the larger the energy increase, the less likely you are to accept it. We also have a temperature factor here, T, which governs, which also affects this probability, and this temperature is lowered over time to make acceptance of sort of unfavorable changes to the solution less and less likely as the simulation progresses. So we then repeat this multiple times, and then once we are confident that we've sort of reached an equilibrium where the average energy isn't really changing much as we swap, we then lower the temperature. And then over time, as the temperature drops, we then explore deeper and deeper into the low energy states, and then we eventually will converge on the solution if our annealing schedule is done properly. The way that we actually go about randomly changing our solutions is by in the following manner. So we first pick a random solution by taking our spin systems shown here and randomly mapping them to the protein sequence. Some spin systems remain in what's called the cache, which are spin systems that aren't assigned anywhere. And then we randomly move these spin systems between different positions on the sequence. We swap them with each other, we move them into the cache, all sorts of different possible maneuvers, calculating the energy and applying the metropolis criterion in each step. And then over time, the solution will converge on a final minimized value that corresponds to the best possible set of assignments. So just to highlight this sort of in a schematic way, we do that random initialization, we perform the Monte Carlo swaps, we check for equilibration. If not, we keep swapping. If we have equilibrated, we then check to see if we should terminate. If not, we lower the temperature and repeat that loop. But if we do terminate, we perform just a quick local minimum search to show that we're at the minimum of the basin that we've landed in. And then we store this annealing result. And we don't just repeat this annealing run once, we actually repeat it about 100 times for each assignment that we attempt. And this is just to make sure that we achieve the same set of assignments, that we're actually reaching the same global minimum. And then we run these consensus generated assignments through curation just to make sure that there are no obviously bad assignments. And then we get a final result here. So this is MVP that Bellagio is assigned. The blue regions are the assigned residues. The white are residues that remain unassigned, and the red are just proline residues, which have no spin system to assign to them. And I'd like to point out that upon comparison, there are actually no mistakes. So this, when compared with Louis Kay's assignments, it shows that we agreed 100% perfect with what he produced. So as you may imagine, the energy function is of prime importance for the accurate assignment. And the energy function itself that we devised is the sum of the energies associated with each individual spin system. And the energy of each individual spin system is itself broken up into two components, the adjacency energy and the chemical shift energy. So the adjacency energy measures the degree to which the I-1 and I resonances correspond with the previous system. So that is the establishment of that connectivity that I talked about earlier. That's how the algorithm handles it. And likewise, the chemical shift energy measures the degree to which the resonances of the spin system match the predicted resonances of the assigned residue. And the adjacency energy itself is very easy to conceptualize as what we do is we just take for example, each resonance pair, C alpha, it's I-1, C beta, and C carbonyl, and so forth. And what we ask is, well, how different is the C alpha from the C alpha I-1 of the next residue? And what we find is that if they're, and what we then do is we say, okay, well, if it's 0, it will fall in the basin of this distribution at a minimum. So that will give a very negative energy. So that's the best possible scenario, best possible match. And as the difference increases, you move towards different out onto the tails here towards a maximum. And then we just sum that up for each resonance pair. So we calculate this Gaussian for C alpha, C beta, and carbonyl resonances, or theoretically any resonance pair that you might want to supply. But really what puts the Bayes in Bellagio, as I like to call it, is the chemical shift energy. And the way that we do this is we calculate the chemical shift energy from a posterior probability determined via Bayes' theorem. And in order to show how Bayes' theorem is applied in this circumstance, let me define 2 events. Event A sub nm is the event where spin system M is correctly assigned to sequence position N. And event B sub M is the event where chemical shifts of spin system M are observed. And from this, we can apply Bayes' theorem, where our goal is to calculate the posterior probability, which is the probability that the assignment A sub nm is correct, given the chemical shifts that we observe for spin system M. And this is equal to the product of these 3 probabilities shown here on the right. And the way that we calculate these, I'll just go through very briefly. So the initial is the prior probability of the assignment being correct. And you can think of this as what would be the probability of you taking a spin system and randomly assigning it to a location on the protein sequence. And that probability is just equal to 1 over n, where n is the number of non-promine residues. And that's easy to conceptualize because you have one spin system and n places to put it. The probability that any one of them would be right is just 1 over n. We then calculate what's termed the likelihood, and you can think of this as a goodness of fit. So what's the probability of us observing the chemical shifts that we observe given the assignment that we're examining? And this is calculated as the complementary cumulative distribution function of a chi-squared random variable, which itself is calculated as the sum of normalized square residuals between the observed and predicted chemical shifts of each resonance in the spin system, normalized by the prediction error. And so here's where we apply structure-based predicted shifts. For this study, I used ShiftX Plus because it has some of the narrowest errors, especially in the carbons. But you could also, if you did not have structure-based predicted shifts, you could use BMRB, general residue statistics, which don't quite have as narrowly defined ranges for each of the predictions. And then finally, we have the marginalization. And the marginalization is the sum of the product between all of the likelihoods and the prior probabilities. And this is really taking into account the fact that even though the likelihood shows that a given spin system might fit well at a particular location in the sequence, it's quite possible that it could fit well at multiple locations. And so that needs to be penalized somehow, and that's penalized by dividing by this marginalization quantity. So now that we have defined all these terms, and we've taken the product of all of them, we can then calculate an energy from this posterior probability. And the energy here is defined as the minimum of this logarithmic function and a user-defined maximum energy. This factor here, K, is merely just defines the minimum energy. What really matters here is the logarithm. So it's the logarithm of the posterior probability over the prior probability. And the reason why I chose this function is because it actually has some very nice properties, mainly that at a maximum probability of 1, we reach our minimum energy, which is our most favorable. When the posterior and the prior probability are equal, we assign it, we give it an energy of 0, which basically means that the chemical shifts data that we provided neither increases nor decreases our confidence in a particular assignment given over random chance. And then in the case of 0 probability, which is the lowest probability possible, we assign it the maximum energy, so it's maximally unfavorable. So now that I've given an overview of how this algorithm works, I'm then went to test it against a suite of 6 test proteins ranging in size from 17 to 61 kilodaltons. It's important to note that this protein down here is actually a homodimer, so its sequence length is shorter than you would expect for a 61 kilodalton system. And the data that we acquired, most of this data ourselves, but for our larger protein systems, we actually got this through correspondence with the Freeman Lee labs, who also provided us their manual assignments. And overall, these proteins were assigned to relatively high levels of completion. So we're starting with fairly complete data sets in this case. And all data sets had full cross peaks for, C alpha, C beta, and carbonyl resonances. So we collected a full battery of experimental data was collected on these. Just running this through using the predicted chemical shifts run through Bellagio, you can see that we got a very high percentage of matched the manual assignment. In some cases, it was perfect. And in other cases, we were missing a few. So we defined missing as assignments that were made manually but were missed by Bellagio. And we also had almost no, only very few cases incorrect assignments, which are defined as assignments that Bellagio made but did not match the assignments made manually. And you can also see that the time it takes to actually run this is actually quite short. So this really can be done in less than 10 minutes on a laptop computer. So no fancy hardware needed to run it. It's just very quick and accurate. The next thing that we wanted to examine was how robust it is in the face of missing data. So the cross-peak lists that we were provided were relatively, that we gathered were relatively complete. So we then took the MPP data set and then we asked, okay, well, let's discard some of the data to simulate conditions in which we're missing large pieces, like large pieces of data, maybe even entire spin systems. So here I reran Bellagio after discarding, or in this case, retaining only 60% or 80% of the total spin systems. And you can see that we were still able to assign, in the 60% case, 55% of the possible. So that's pretty close to the total possible, which would be 60 given our data constraints. And similarly, we got to almost complete assignment with 80%. Likewise, we then examined just individual experimental pairs. So we just looked at C alpha resonances, keeping the C alpha resonances and running that through Bellagio, just the C beta resonances and the carbonyl. And you can see that in the case of MBP, we're actually able to get almost complete assignment with just the C alpha pair alone, or just the C beta pair alone. But we ran into problems with the carbonyl pair, which we expect have to do with the fact that very little residue type data is contained within the carbonyl. So it's pretty ambiguous as to where that gets located. And then finally, in terms of this testing that we ran, we looked at just discarding individual resonances. So we then discarded, or only retained rather, 88%, 50%, and 75% of these various carbon resonances that's shown in condition 1. And then these were some other conditions that were run in a similar manner. And you can see even with severe missing quite a bit of the C beta and carbonyl resonances, we were able to get pretty close to complete assignment. Only in this condition did we dip below 95%. the total possible. But we really also wanted to test this a bit further. So we wanted to take a more challenging protein system. So we used the data that was provided to us by Dominique, his 52 kilodalton Cy1 protein. And we then just looked at individual individual resonance pairs, similar to what we did with MBP. And we also tried different subsets of the resonance pairs. So what we found was we were able to assign, just with the C alpha pair alone, almost 80% of the protein with a relatively low error rate. But this error rate almost completely vanished when we included the carbonyl pair, and the completeness of our assignments rose to above 95%. And what's remarkable about this, in our mind at least, is that no C beta data was needed to actually assign this, which is the least sensitive data, which is the least sensitive of the triple resonance experiments. And furthermore, because when Dominique assigned this protein, he didn't have a structure to go off of. In fact, he just wound up solving the structure. We tried that ourselves by relying solely on an AlphaFold predicted structure in order to generate these predicted chemical shifts. And we found that even with an AlphaFold predicted structure, we were quite successful, which shows that even lack of an experimental structure isn't that much of a barrier. So in conclusion, Bellagio is fast and able to assign large proteins, even with sparse data sets. And that we can use predicted chemical shifts from AlphaFold structures in lieu of a experimental structure. Where we're going with this next is that we really want to combine this with automated peak picking, because Bellagio, we would think it would be great if Bellagio could inform on the need for additional assignment data in real time, so you could stop collecting experimental data once you have enough to complete the assignments. And furthermore, we'd like to experiment with augmenting the energy function to include amide, and OE information, just to help in cases where the data might be even sparser than normal. And we're looking to make the software available for general use on NMR Box. And with that, I'd like to acknowledge the people who helped me with this project, notably Sravia, Gladice, and Kyle, who helped me collect the experimental data used for the assignments, as well as Dominique and Andrew for their protein data sets, and our funding sources at Mathers, the NIH, and Texas A&M University. And with that, I'll take any questions.
Thank you very much for the presentation. So I remind everybody, please use the Q&A box for putting your questions. So we already have one from Jeff Ho. Jeff says, really nice work. Did you compare your results with the performance of other packages, Abacus or Pine, or if I may add Mars? Looking forward to getting it on NMR Box. And I could add there, I mean, the comparison would be probably good for, in the absence of predicted chemical shifts from a structure, to really see what is going on.
Yes, so I can answer both of those. So we did test against Mars and iPine. And we found that overall, if you look over here on the right against our test suite, is that Bellagio was quite successful in generating the assignments. Pine tended to have considerable error rates for one of our protein systems. And in addition, the larger protein systems, we ran into trouble with Mars, in that it was having difficulty. I don't know why exactly, but we were unable to really get it to assign our two larger protein systems. And what you'll notice is that I omitted Pine from some of these, because Pine actually searches the BMRB database for using PAXI. In order to help the assignment process, and so I omitted the proteins that were present within the PAXI database at the time of the assignment. Although I would like to point out that I did run Pine anyway, and Bellagio did have a slight edge in performance over those, but it was pretty negligible.
Could I follow up there, maybe? Just, is this with predicted chemical shift from structure or without?
This is with. so Mars, I used predicted chemical shifts as well. The predicted chemical shifts from BMRB, let's see here. Sorry, unfortunately, I don't have them. But yes, the performance, in the cases of sparse data, the performance degrades considerably. In fact, actually, I have that. Where do I have that? I guess I didn't include it. My apologies. But yes, the performance degrades considerably without predicted chemical shifts in the cases of sparse data. But relatively complete data sets, it performs quite well.
Thank you. So then there's a question from Daiwei Lee. What will it perform if you have lots of noise peaks?
Yes, so Bellagio, there's no sort of filtering for that sort of data. So I would expect that Bellagio's performance would suffer if there are a lot of, if you're picking resonances that aren't really there, would be my prediction, but we haven't examined that directly.
Okay, thank you. Then actually, Bex has a question. Very nice work. Would your program be able to handle cases where separate distinct conformers are present, such as may exist if a protein contains cis and trans pro-conformers that extend over many residues?
Yes, so the algorithm allows the user to specify which spin systems they believe are actually derived from the same amino acid. And it considers both spin systems that you observe in your triple resonance data when making the assignments. So it doesn't make the judgment itself, but you can tell it that you think that these two spin systems belong to the same residue.
And there's a question from Tobias Schubais. What is the input, just peak list, or do you need to assign peaks to spin systems manually before?
So right now you need to build the peak lists, I'm sorry, the spin systems yourself, but we're moving towards automating the peak list, or the, I'm sorry, the spin system construction from peak lists.
Okay. Then Angela Cronenborn, when you prepared spare sets, did you remove at random? In real life, there are missing resonances are usually not random.
Yes, so we removed them at random, and we were considering what sort of the impact of that would be. And in some ways, having all of your resonances missing from the same region is actually kind of favorable in a way, in the sense that you're able to maintain connectivities in the other regions. But I suppose it's pretty case specific as to whether or not that is actually to your benefit. Because if you figure if you're missing a relatively few and it's scattered over, that's sort of the best situation because there's enough data still present to figure out where everything belongs.
Okay, thank you. Then Alexander Klein, nice work. Do you plan to add some customization like new other correlation experiments?
Yeah, so the way that I'm doing rewrites to the algorithm right now, and what I really would like to be able to, where I would like to go is be able to have it be sort of very general, where the user will be able to specify any resonance pair they want to establish connectivities between. So you could have any arbitrary I-1 resonance pair that you could supply to it, and as well have any arbitrary predicted shift that you want to compare it against. So more or less the user would be left to define what they want their input data to be, and Bellagio will sort of figure it out, what needs to be compared to what.
Okay, I think these were all questions in the Q&A. So there are two more questions from the panel, but I would close the official session and then we can go to the unofficial and start with the panel member questions. So thank you, Anthony, again for the very nice presentation, and also Roberto, and then we go to the unofficial part and I stop recording.